Yeah, good morning, everybody, and welcome to the second lecture in microbiology. Yeah, this time it took a while. I'm sorry for that. Um, it looked like that I messed up the first stream because I changed the latency. Pressing the button start the stream, I changed the latency, and I think that the software didn't like. Unfortunately, then you cannot stop the stream and restart it because this will simply stop the stream and end it. So I had to set up a new stream. However, you need to do that, of course, in parallel with a running stream. And therefore, with two browsers and so on, close to a nightmare. It worked, but hopefully next time I will never ever hit while starting the streams any buttons again. Promised. So I also promised last time. You remember we had a very long session, and at the end it was maybe a bit hard because it was so condensed. The reason was simply because I wanted to end the complete Corona package because I wanted to extract that, as you have seen it, as an own video, which is now free on YouTube, because I think that this one is also interesting for other students, and maybe even not students, and therefore I extracted that, and therefore I took a bit longer. As we started a bit later, also today, it will be a bit longer, but just based on the starting point. Maybe also we can end in time today. So again, sorry for this problem with the stream. Thanks, Ralph, that you announced immediately that there is a problem. So maybe next time I also learn what I do is, Ralph, I will send you a message, stream started, and if there is nothing coming in your hometown after a minute, then we know there is an alarm and we have to change something. Maybe for the future, the problem is with the chat. If I stop the stream, the chat is gone. So if there is a problem with the stream, please go back to Moodle. So I will then put the new link in Moodle, which makes life for me very much easier because I can stop one stream and set up a new one. So I will put that also in Moodle as a text that link addresses might change during the streams. If there is a problem with the stream, that then you have to refer to Moodle in this case. Hopefully in the future this will not happen anymore. It happens the first time, but things happen. Okay, so let's go to our question, which was a real difficult one, of course. The question was, what statement with respect to antimicrobial drugs is not correct? Now I just wanted to check. It's not many people here, so maybe Ralph, you could send me a WhatsApp message to you. You put also this link to the old stream. That's important because that's where the people can change it and Moodle. And please to the students that you put it. Put the new address also in your WhatsApp groups or whatsoever, in order that the people have a chance to get to this stream. Okay, so now coming to the very difficult questions. So that's what I want to do always now, a question at the beginning. Maybe the future question might be a bit more difficult than this one. So let's have a look at the question. The question was, what statement with respect to antimicrobial drugs is not correct? Chemical modification can expand the activity of penicillins to gram-negative bacteria. Oh yes, of course they can. So therefore you generate ampicillin, for example, from penicillin using a modification, which is, I think, just an amino group, and then you have the passage is possible through the membrane. Glyphosate antibiotics are rarely used as they're toxic and high resistance frequency like kanamycin, and beta-lactam antibiotics essentially act by interfering with cell wall synthesis. Of course, listening to Trump makes you look better, maybe. But for me, the real, really the incredible thing was in the morning I told you about this chloroquine stuff. By the way, also Elon Musk. I'm more a fan of Elon Musk, I have to confess, than of Trump. But also Elon Musk said use chloroquine. No, yeah, even if it's a Musk, also in this case, it's not true. But in the evening, when last week we had this session on Corona, in the evening Trump went out and said the people should inject disinfectants. This is this is this is really, you see that, and you think, what? 20 people were killed, most likely, due to this sentence. Maybe that's the possibility to send him to prison because he merely killed the people using such an approach. That's really incredible. I could uh, um, shake my head for hours for such a thing. Okay, but now we come back, and again, today is way more relaxed, and I have a look in the chat. Oh, I have to change the chat address. I don't think that I have done that already. So give me a second, because in Moodle we have the, not in Moodle, but in here we have the old chat. So everything is in principle prepared. So this should be now the correct one. Okay. So now back to the board. Here we are. So just looking whether there are questions. He made a joke. They couldn't be. That's true. That's also what is my opinion. It's not his fault. It's always the fault of others. I had received some questions with respect to the points. I just remember. Now for you, it's clear there is no lab course, so therefore you will have the lab course in whatever spring or summer next year as a block. There will be the homework, so you write the exam as normal, and then you will get the complete module accredited as soon as you have the lab course. So that's not a real problem. It's just let's say that it's then delayed, that you get the credits for the course you can only get after you have finished the lab course. So there are people who have done the lab course before from higher semester. So these people, of course, will get the lab course already accredited, so they don't need to do the lab course again. However, the points of this lab course will not go into the exam, so it's just then the homeworks and not the points of the lab course. I think that's it for the questions with respect to this topic. Again, if you have additional questions, if I have forgotten something, just tell me. Okay, so now let's go back to the presentation. I see it's just 34 people. I think that's really my problem, because I messed up the streams again. Sorry for that. Next time, hopefully, this will not happen again. I promise. Okay, so coming back to the topic here. Now, we have talked last time about antibiotics, how they work, and we have talked about antiviral agents, and of course I have already written it. The section on the complete section on antiviral agents is not relevant for an exam. In whatever type the exam will be written, this is not relevant, but it is of course actually relevant because we have the corona situation actually. Okay, now let's go here and let's talk about antimicrobial drug resistance. So we have the problem that many drugs don't work. We had talked already with kanamycin that we have many resistant bacteria, but the resistance comes up also for others. And of course, the question is, what is the reason? What is the mechanism behind? So there are several mechanisms which can confer to bacterial resistance. So although there is no little man here, it's clear, don't learn this list by heart. It could be that I asked you give an example of a bacterial resistance mechanism and then you give an example, and then it's fine. But you should have an idea of how this works. So there are, you see on the left side, there are the mechanisms. So some of them are, they're depicted here: reduced permeability. So in this case, the antibiotic cannot penetrate. For example, in gram negatives, this penicillin, normal penicillin, doesn't work. So that's all gram negatives have that. Then antibiotics can be inactivated. There are several ways to do that. You remember that for penicillin, it's the beta-lactamase, which just cleaves the beta-lactam, and then it's no more active. But you can also methylate, acetylate, phosphorylate, and so on in antibiotics, and then it's no more active. You can also, if you have antibiotics which attack a certain target like ribosomes, you can, or the RNA polymerase, you can of course also alter the target, so mutate the target in a way that the antibiotics doesn't attack it anymore. So in this case, it's of course based on a chromosomal mutation. It's logical because these targets, RNA polymerase, ribosomes, and so on, are essential genes normally, and they're not plasmid encoded. And for antibiotics, inactivation mechanisms like beta-lactamase and so on, where you have an enzyme altering specifically the antibiotics, it's very frequent that you find the information on a plasmid. And I would say this is kind of why the plasmids were invented, because then bacteria could, via horizontal gene transfer, quickly transfer the resistance mechanism to others. Resistant biochemical pathways is of course also a possibility. That's the case for sulfonamides, and what's also there is just efflux pumps. So the antibiotics gets in the cell, and at the same time there is a pump pumping it out. This is a typical resistance mechanism in eukaryotic cells, for example. So in cancer treatment, you observe that very often, that there are certain generic pumps in eukaryotes, including humans. And if you have a treatment of a disease like cancer, these pumps often get upregulated in cancer cells, so that the initial dose is no more effective, and you would have to increase the dose. The problem is, of course, sometimes that you have a certain therapeutic window and you cannot increase doses indefinitely, and then you will have a problem. Okay, so this is just a summary of resistance mechanism. This is how this could look like. So phosphorylation or adenylation in the case of streptomycin acts at this hydroxyl group. We have already talked about the beta-lactamases, which can inactivate penicillin and cleave at this position. It's like the peptidic link, which is then cleaved. And you have also, for the case of chloramphenicol, the example is that you have an acetylation here. Okay. Maybe one, as I see Brock biology of microorganisms. We have a real problem, because I have asked at the library whether the book is available, could be made available online. There is this Brock cannot be purchased in a online version. So this means that for this year, and Pearson is, I have also asked them whether it's possible to make Pearson in general available for students. So there are models. I will insist again, but I don't see a real chance, because we have licensed in, just for you to have an idea, we have licensed in Springer, complete package of Springer, all books. Unfortunately, I have looked, there is no microbiology textbook for Springer. So they're more specialized textbooks. So there are microbiology textbooks, but then they're always wine microbiology and so on. So it's not a general textbook like the Brock microbiology. There is for Pearson and other, let's say, things like Elsevier and so on, they usually offer the libraries also these licenses to get all books, but they're really extremely expensive. So that's hundreds of thousands of euro for the library to license such a package. And this is our problem. We don't have this money. It's very simple. So something, I look deeply into your eyes and I tell you something. So if you look at the Brock microbiology, you will see that if someone, whomever has this, and then can sell it after this course again, usually there is a link to the to an online version of this book. And I don't think that you can distribute the link. But I know what I do here is I register the screen. I'm silent now. So that's something you might need to think of. So if someone has this book, it might be that there is the possibility to get that others also can get access to this book, and you might share the workload in this case. That's just a suggestion. We as a university, unfortunately, we cannot change the situation here because that's Pearson, and there is we cannot license single licenses for this book as ebooks, because it's not available. But in this book you have a code. If you buy this book, you have a code where you can have access then to an online book, and this is something which might be possible to share. And I don't exactly know, but it might be that I find an old version of a PDF version of a older Brock book. If I find this, it might be that this finds the way to Moodle. It might be, yeah. So this is it's a pity. I'm sorry for that, and I know that the situation is not ideal. However, in this course, it's certainly not easy without a book. But you found most of the information you will find also in Wikipedia and so on, although that's not optimal. So we recommend, of course, the books. But therefore, please take the chance in the lecture to ask and so on. So you feel free to ask. Also, feel free to ask if you had a problem with understanding something from the last lecture. Put it in the chat, ask. Yeah, so this is really no problem. And for the people who sit in the States or in America, where the time zone is not very ideal, send me an email. Address on the email. You sit in this time zone, you have no chance to get to the lecture. So then I can also ask or say that during the lecture, and you have then of course later access to this lecture, as you know. So this is something I could offer. And maybe you could send someone else, just as a safety measure, a message that he or she asks for you, in order if I forget it, which could happen, because if I mess up the streams at the beginning, it could well be that I forget then something, some of your questions I have received via email. So it's always better to put these questions in the chat, because I will see that. I will look from time to time in the chat. So that's what I do now. Let's have a look in the chat whether there are new lab course script. So there is a lab course script on Moodle. You can download it or not, but it's not really makes no sense. Edition, the current edition is the 15th edition, to the best of my knowledge. Side effects of chloroquine phosphate. Why we can't use it. So hydroxychloroquine has side effects. One side effect is QT prolongation, which is a cardiac side effect, and usually it's combined with an antibiotic, and this antibiotic has the same side effect. So you also always have to think of that the side effects not necessarily need to be severe. You know that chloroquine is used as a malaria prophylaxis, but usually this is taken by people who are quite healthy. These are not the people who are really affected by corona. In general, people in your age with 20 years are not on the intensive care unit. There are always some exceptions, but in general not. So the people who are at the intensive care are usually people who have previous diseases, often also cardiac diseases, and if you treat them with a drug like chloroquine, you expose them to a real risk dying of a cardiac side effect. Just as an example, if you read the side effect lists of these drugs, it's usually not this line, it's more than that. And it's not very smart if you have not yet tested the drug in a certain indication, to give a general recommendation to use it. That's the problem, because you don't know whether it works. And as I said last time, I have seen many, many drugs failing in phase 3 trials despite that they worked in early trials. And this is simply because these people have no experience in drug development. So you need to have the clinical trial first. Of course, you need to go for the clinical trial. So if you have a hint that chloroquine works and this is an approved drug, people will do studies to see whether it works, and that has been done. But now the data is out, and obviously chloroquine doesn't really work. So that's the reality, and of course it's a pity. I would like to say here chloroquine works. It's not that I want to say Trump is not right. I want to say it's not okay if there are hints that it could work, for example, based on the mode of action, to go on a general basis to expose people to a risk of the side effects of such a drug if it's not proven that it works. And at present it looks like it has been disproven, but people don't see that. So people even use in the States, people have started to use chloroquine. Healthy people indeed used chloroquine because they were afraid to listen to Trump, and then they started to take the drug, and they came with intoxications into the hospital also based on chloroquine, and that's a disaster. That's a nightmare. You should not do that. This is not something. If you're president, this is impossible. This is just impossible. So that's the point here. Okay, which edition is it? Yeah, this is, I think I have said it. Okay, now let's come back to the slides and let's go to the next one. So this is just a very sad slide. This is an appearance of antimicrobial resistance in pathogen, and this is the correlation between use and resistance. So the more you use in tons, the higher the frequency of resistance in general, with some exceptions. So you see kanamycin is an exception, but you see that this curve straightly goes up. And you have here some strains where I have put these exclamation marks, where you have isolates which are resistant against all known antibiotics. This is a nightmare, because if you get infected with these strains, then you cannot do anything. So you have this bacterial infection and you cannot treat that. So Pseudomonas aeruginosa, for example, then forms in the lung. It's not very, usually if you're healthy and so on, you don't really have a problem with this strain. But if you have a lung disease, for example, cystic fibrosis and so on, this guy starts to form biofilms in the lung. You cannot treat that really, and people will die due to this strain because you cannot treat it anymore, because there are no more antibiotics, and this is of course a nightmare. Okay, now this is just the, let's say, the background how you can come to resistance. But in the clinical setting, of course, it's important that you know how whether or not an antibiotic is effective, in order to know what antibiotics to use. So one important measure here is assessing the minimal inhibitory concentration of an antibiotic in order to inhibit the growth of the test organism. So this is done by a tube dilution technique. I want to show you briefly how this looks like. So in this case, what you do is you have your test tubes. Oh, red is maybe not the ideal color. Let's go to blue. So you have your test tubes, and now you simply do a dilution series of your antibiotics, and you might have here a concentration of whatever, 10 milligrams per liter, 1, 0.1, 0.01, and 0.001. Yeah. So, and now what you do is you inoculate with your test strain and incubate it. And then what you observe is or maybe you, as a control, you also put 0. So, and then you look where your microorganism has grown. And then, of course, at 0 it should grow, otherwise you have a problem. And it also grows here, and it lightly grows here. Here there is no growth, here there is no growth, and here there is no growth. So this means that this concentration here was sufficient to block growth, and therefore this concentration here in this case is the MIC, the minimal inhibitory concentration. Okay, so this is how this MIC assay works like. So in brief, you make a dilution series of your antibiotics, have then different concentration in a medium, of course, in a growth medium. Then you inoculate with your test strain, and then you incubate, and then you look up to what concentration you can completely block growth. So what you measure here, or what you look at here, is simply the turbidity of the flask. This is a very this assay, to do this assay, it's quite easy, so it's not really a problem to do this assay. So this is widely used, and you can easily then assess that. Okay, so that's the MIC assay. It's very important to understand that it's a block of growth. Yeah, what you see here is simply that you block growth. It's not killing, it's block of growth. And it's important, if you do an MIC assay, you must standardize the assay. So you cannot compare the results if someone else has taken different inoculum concentration or different strain, a different temperature, pH variation, and so on. So these, it's of course it depends on temperature and everything. So it's important that you have standardized conditions. You will see in a couple of slides how important these values are and how they can kill your experiments. Yeah. Okay, now another method is the so-called diffusion method. This is more simple. So what you can do is, in this case, you inoculate discs, filter discs with an antimicrobial agent. Then you place it on a plate where you have your test organisms, and then you incubate, and then you check the area around this discs, and of course the diameter of this area of inhibition is proportional to the activity of this agent. Yeah, so you can then somehow also quantify the inhibitory activity, and here you can of course measure different concentrations, or you can measure different agents at the same time on one plate. So that's a quite quick assay in this case. Okay, now I want to show you an example how that could lead to problems and what problems you can experience. And the learning from the own errors is the best. So this is an example of a PhD thesis. So this money I got the grant. This was a DFG grant, and this is Katharina. She was my PhD student doing the job. So that's, by the way, Katharina at the Kinder University. This is not for doing this project. So that was still during my time in Würzburg. That was a joint graduate college of the MSZ in Würzburg with the University of Nice in France. So the project, as I said, is called Caspase-1 as a target of bacterial tumor therapy. What I want to point out here is not the complex project. I want to just show you where we have a problem with these antibiotics, inhibitory concentration, and the antibiotics we use here is gentamicin. Now, just that you can understand, of course, everything we have here now is not relevant for the exam. So what we have here is that we have a tumor with tumor cells, and the point is, a tumor usually doesn't look like this. So it's not just tumor cells. So you have other cells in the tumor also, like fibroblasts, endothelial cells, and, for example, tumor-associated macrophages. And we wanted to know if it was known at that time that if you inject attenuated bacteria into a mouse which has a tumor, it's also known that it also happens in humans, where actually do these bacteria go to? Because we used strains which could infect tumor cells, so facultative intracellular bacteria like Salmonella, Shigella, and so on. And we wanted to know, they're attenuated, we wanted to know where do they sit? Do they sit outside of the cells in the tumor tissue? Because we also work with completely extracellular bacteria and they go to the tumor. Do they go into the tumor cells? This is something what you wanted to do because this is a kind of tumor targeting, and then you could do things in the tumor cells. Do they sit in other cells or do they sit in macrophages? So there are also macrophages in the tumor. And this is what we wanted to answer in this project and what we did answer. Now, of course, technically you should do that somehow. So the first step is you excise the tumor and make a total cell suspension of your tumor cell. This is technically not so easy, by the way, yeah? Because you need to have single cells which you can analyze. So there, I have to say, Katharina did a great job. There was a technically excellent work, really technically excellent work. And now you want to know, where are these bacteria? So you have one thing is measure total bacteria. So you have your total cell suspension, and you just plate everything. And of course, if you just plate it, then you will each time where you have a growth of the bacteria, you know, okay, that's one bacterium, fine. Now, if you want to, then you want to see how many bacteria are in the cells. So you cannot wash that. You have to kill bacteria which are outside of the cell. And in this case, we added gentamicin. And gentamicin is an antibiotic which doesn't penetrate into eukaryotic cells. So this antibiotic stays outside of the cell. So therefore you kill bacteria outside of the cell, and the bacteria inside of the cell stay alive. So with this, we get then the bacterial counts which are inside of the cell. Yeah? And then in parallel, what we did is we separated macrophages from the fraction where we have no macrophages. So we separated the cells. We used then, this is magnetic sorting, we used then antibodies detecting markers on the macrophages coupled to magnetic beads, so we could separate these macrophages, and then we had macrophages and macrophages depleted, and then we analyzed where actually are these bacteria. Are they in macrophages or are they in the macrophage depleted fraction? So that's the setup of the experiment. By the way, you can, of course, you have to show that you can purify the macrophage, so we have a very pure fraction. These are macrophage markers, F480, CD11B, and you see the fraction is very pure. You have 97% of the cells here. That's a FACT staining, and the remaining fraction is also very pure. You hardly find any macrophages there. So, and this was the, let's say, the number one experiment here. So we injected, we have mice bearing a breast cancer in this case. These breast cancers have high numbers of macrophages inside. And we injected then salmonella, an attenuated salmonella strain intravenously, and then we looked where we are. And you see that's 4 hours after injection, 6 hours after injections, and 7 days after injections. And I want to come to the gentamicin, so I didn't forget it, that I want to show you the gentamicin stuff. Now, you see that this is spleen. So spleen plus minus genta, you see that there's a high amount of bacteria getting into the spleen. This is a log scale, yeah? And so at the beginning, that's normal. You would expect to have many bacteria in the spleen. After 7 days, these bacteria are completely eliminated in the spleen. So that's also normal because these are attenuated bacteria. But this is not the case in the tumor. You see, even after 7 days, you have these highly attenuated bacteria still there. And now if you look here, I just want to look at the 7 days. So here we have this total cells minus genta, total cells plus genta. So this is without gentamicin, and this is with gentamicin. In other words, these are the bacteria outside of the cell, and these are the bacteria inside of the cell. And you see here you have around about tenfold more bacteria outside of the cell than inside, which is interesting because at the beginning they were all inside. This is something which we could talk hours about, but now I want to come to this point. You see at the beginning especially that you have way more bacteria in the macrophage section. So here you have still a higher amount of bacteria in the macrophage fraction and less bacteria in the macrophage depleted section. Okay, so everything looks good, but then I had a closer look at this bar, at this chart, and realized there is something wrong here. And now I could ask you what is wrong here. This is certainly not so difficult, not so easy to see. However, if you go back here, of course, the cellular fraction here, what you treat is always treated. You want to see here, you just want to see intracellular bacteria, because here, of course, you don't want to see bacteria outside. You want to see whether the bacteria are in macrophages or they are in the macrophage depleted fraction. So here you always treat with gentamicin. It's not on the chart because it's clear that you treat with gentamicin. So in other words, this is treated with gentamicin. This is the total cells. This is the macrophage fraction treated with gentamicin. And this is the macrophage depleted fraction treated with gentamicin. And the problem is that both of these bars, so the fraction, both fractions have more bacteria inside of the cell than the total fraction. And this cannot be. This cannot be correct. This bar must be somewhere in between. If this happens, it means that something with this experiment was wrong. So we did a complete experiment, and then we ended up with a figure where we said there is something wrong. We have to repeat everything. And of course, at the very beginning, we didn't know what was wrong. So we checked our gentamicin. That's what we needed to do. So so we had the values we used, we had from literature values. So in literature, they used 100 micrograms per ml gentamicin to kill other bacteria. To kill, not to block growth, to kill. So killing is something else than blocking growth. Killing means that the cells are really dead. So an MIC concentration, that's very important for you to remember that. An MIC concentration is a concentration where you inhibit growth. It could be that you kill, but it's not necessarily that you kill. Usually inhibiting growth, you can use lower concentration for inhibiting growth than for killing. So what you really need, first of all, is a concentration where you readily can kill the cells. And now we checked number of bacteria after gentamicin incubation with different concentration and an incubation time of 1 hour or 2 hours. So it's a kinetics. We need to kill, of course, the bacteria in a certain time. We cannot incubate the cells for 5 hours with our gentamicin, then our cells are dead or lysed. And you see here that with a 50 micrograms concentration after 1 hour, you have hardly any activity, and the 100 microgram concentration, you have an activity after 1 hour, but it's around about maybe tenfold. This is for us not enough. We need more. So we need to have at least, you see that the differences in the cell and non-cellular fractions are already 2 log scale, so they're 100-fold. So we need at least a 100-fold or a higher activity in order for the killing. These values you don't find in the literature. The people just use it. So we switched to 300 micrograms gentamicin because there you see you have a 3 exponents reduction, and that's fine. So it's a 1,000-fold reduction, and that's for us fine. Of course, you could incubate even longer, but it's a problem to have the cells 2 hours on room temperature or 37 degrees. And you see that At these higher concentrations, this is a linear scale. It's not a difference with respect to killing intracell. It's an important control to do that, because if you increase the concentration, it could be that you have also an effect on intracellular killing, what you don't want to have. You don't care too much about the cell, so we didn't check for cell viability. We just wanted to see whether the cells inside of the bacteria inside of the cells are still protected. Yeah, so that's the important thing. So this means we incubate the whole cell suspension with 300 micrograms gentamicin for 1 hour at 37 degrees. That was the new protocol. And in parallel, we perform a max cell separation in the presence of 300 micrograms genta on ice. Because this is the staining procedure, it has to be on ice, and then we determine the CFU. Then it turned out it didn't work either. And now the problem was the temperature. One was incubated at 37 degrees, the other was incubated on ice. And now we compare the incubations with gentamicin 300 micrograms per milliliters at different temperatures. So this is the control without gentamicin incubation, and this is the 37 degrees, and this is the 4 degrees. And you see at 4 degrees, gentamicin has virtually no activity at all. In other words, if we incubate just it on ice, there will be no killing at all. So we had to repeat the complete in vivo experiment and perform the cell separation after 1 hour pre-incubation at 37 degrees, because the data was not solid. And then we got it. We got the same at the end. We got the same. More or less, the figure looks the same, just this bar was here. And it was just a bit shifted, so the take-home message of this experiment was the same. But it's science. If there is an error in the experiment, you cannot use that. You have to repeat that, because it's not solid. This is very important. And of course it was a pity, because it is an animal experiment. This took a couple of months to repeat this experiment. It's not something what you do in 5 minutes. So this was, of course, for us an experience we would have liked to miss. But the good news is that you know that now, and hopefully you will not make the same experience on your own. Okay, so that's about this. Now I just have briefly a look in the chat. Won't there which additional for MIC treatment increase the number of dilution steps to find a more accurate value, up to which point it's really useful? What does the NS mean? Maybe what's a good idea is, if you have questions, always refer to the slide number, because then it's quicker for me to find it, because I don't know, of course, what in S you mean. Won't there be a concentration between 0.1 and 0.001 or 1 and 0.1 where the actual MIC might lie? Yes, but so you're right. It's not accurate in the way that you could say it's the MIC is 0.37334. Two things. Point one is it doesn't matter in this case that you have a real exact value with 5 digits after the comma. You want to know in what range you need to come first. Second, you can do it more accurately. What you do in this case is it's not helpful to do a dilution where you say it's I do 0.1, 0.09, 0.08, 0.07, and so on. Usually this is not how you do it. Usually you would do, because then you have also the statistics, then you have that maybe if at 0.09 you have no growth, and at 0.08 you have growth. Now what? So this is always a certain fluctuation. What you can do is okay, now I just come back to the NS first. So this means not significant. So this is, of course, we tested statistically whether this reduction here is significant. This reduction is not big, so it's a 50% reduction from the from this 50 micrograms to the 100, 200, and you see here it's not 50%, it's 1,000-fold reduction, just to get the scales into theory. But this reduction from this to the others is significant, and if we measure this to the 300, because that's the concentration we want to use, from 500 to 300. But from 100 to 300, and this is the concentration we have used at the beginning, it's not significant. And it was important for us that it's not a significant, it has not a significant effect of the intracellular bacteria. We don't want to have the cells alive, but we want to have the cells, the cell integrity kept, that the gentamicin cannot attack the intracellular bacteria, first of all, and second, we need to be able to do a max separation, and also there we need to have an intact cell integrity. Then later on we plate the cells on agar, and this is something they will not survive, but there we don't care anymore. So we don't need to keep them alive for the complete procedure. Okay, now I want to go back to this slide. So what you can do is, if you wanna have a quantitative information, instead of going for this this 0.09, 0.08, and so on, what you can do is, and what you should do is, do these approaches in parallel. So do it again, and again. So do that, for example, 20 times. And then you can, by statistical methods, simply calculate the more precise value, and you have a statistical variation in this case. So that's better to do that. And of course, if you know that your drug is active in the range of 0.1, you don't need to do a complete series starting from 10 milligrams to 0.001 milligram per liter. This is something you would do just if you have no idea of the activity. If you know that the activity is in this region, you would just take maybe from 0.1 to 0.001, for example, in order to pinpoint it. Or there you could also make just a 3-fold dilutions instead of 10-fold. But it doesn't help you too much if you make a smaller scale here, because just about statistics, it could be no growth here, growth here, again no growth here, and then, well, then it's still no 0.1. So that's the problem here. Okay, now we come back to the presentation. So, and we go to the next presentation. Okay, just give me a second. Just checking whether there are more things in the chat. What's the minimum amount of difference you can have where you stop considering it NS? So you're a second semester, so therefore you don't have statistics yet. Statistics is something which is very important. This is something which will be more conclusively answered in statistics, but very briefly. Of course, that's a very important thing. So you can have two things. So at the end, what you want to measure is this is outcome. This is whatever you for example, you do your chloroquine trial, yeah, and you measure then the healing time in days. So this is for example, this is 10 days. So without treatment, you have people take 10 days to recover, plus minus something. So this is the mean plus minus standard deviation. Now with chloroquine, it's 8.5 days. So this is 10 days in mean, and this is 8.5 days. Yeah, this is chloroquine, and also plus minus standard deviation. Now the question is, of course, it is a difference. But now you want to know, is this significant? Is that just by chance, or is that something which came up because there is a treatment effect? And then the significance, mainly depends on the standard deviation. So if you have now standard deviations like this without a statistical test, I can tell you this will not be significant, because these standard deviations, you can prolong that here, will overlap. So it's just also by chance you can get that significance. And what you use, of course, is a statistical test. In this case, you would use a t-test, which is a parametric test, which looks which is a mathematical test, a statistical test, in order to see whether significance is there or not. And then you can quantify that. And you get out a p-value, and this p-value tells you what is the likelihood that you see this difference by chance. And usually you say, if the likelihood is 5% or lower that you see this difference by chance, then you term it significant. Yeah. So, and now it could be that you have something like this, without, with, this is now the difference between statistical significant and relevant. So this is 10 days, and this is 9.3 days. The standard deviations are very small, so this is significant. So if it's significant, you put a star. But then the clinician would say, well, it's significant, but I don't expose my patient to a side effect for just gaining 0.7 days. This is not relevant. Yeah, so that's the problem here. So this is significant, but not relevant. By the way, it was not significant, the difference. Yeah. What was found. And another possibility is that you have this. You have a large difference. This looks very good, but you have very large variations. And also this one will be not significant. And this is often something what you see if your assay, or if you have small group sizes, for example, for these trials, small group sizes, you often cannot give a statistical correct answer. And this is the problem. At the beginning, these tests are just occasional tests, and they often then go into the direction that you test just 5 patients and you see a treatment effect. And this is what this is a certain bias you often see in the clinics, because you want to see a treatment effect. Although you term yourself as being objective, you look at the people different, you act different. And if you do that in a double-blinded way, which is usually done in a phase 3 trial, often these effects unfortunately disappear. Again, this is something what I have seen a lot of times. Yeah. So, and this is the problem if your assay system is very error prone. By the way, if you do CFU assays, you will see it in the lab course. If you count bacterial colonies via spread plating, assess them via spread plating and count, you will see that in these assays you very often see standard deviations in this range. And this is, of course, this kills then your statistics completely. And you have seen the values before from my PhD students. I have never ever seen, and these are all spread plating CFUs, I have never ever seen so brilliant CFU values, which have such a low standard deviation. That's really incredible. That's really good. Double-blind trial, okay, that's just now to finish that. A double-blind trial is a clinical trial where the doctor doesn't know whether he applies your drug you want to test or a placebo. The doctor doesn't know it, and the patient doesn't know it. So a single-blinded trial is the patient doesn't know it. A double-blinded trial is also the doctor doesn't know it. So the doctor cannot give you the drug like this. This will help, or kind of placebo will not help. It doesn't know it either, and this is of course the best way to do a trial. So of course this is now also done with remdesivir. By the way, also unfortunately remdesivir doesn't show the effect what you would like to have. Yeah, remdesivir. Very recent trial came out, I think a couple of days ago. Also, unfortunately, remdesivir does not look as good as at the beginning. I don't want to say that this is dead. I just want to say that unfortunately it's not that if you start treatment tomorrow, that everything will be fine. I think the remdesivir data is based on a Chinese study with 400 patients. The problem is that they have applied it quite late and couldn't get all the people in the trial. So it's not necessarily said that it's not useful, because if you come early it might be useful. However, at least the effects are not so pronounced as you would like to have. That's the truth. I would like to tell you something else. It's not that I want to tell you Trump is a liar. This is that you don't need to wait for trials like this. However, unfortunately, now the drugs which are being tested are not so brilliant. To the best of my knowledge, remdesivir was not approved in Ebola, so therefore, of course, the companies there are quite pushy because they want to bring out these drugs on the market, because that's, of course, a huge market. So that also should make you very careful. Okay. Yeah. So that's the unfortunate answer. Okay. Now, as promised, I said that we start with infection biology, and we will talk in this section. The major part of the section will be on bacteria. So, because I would say viruses, I have already talked about viruses last time, so you have seen the infection cycle of the coronavirus. However, bacteria have their own metabolism, and I'm coming from bacterial infection biology. I don't want to say that it's more important, but I would say you can learn more from bacteria than from viruses. Yeah. So at that place. So therefore, we do most parts with bacteria. We will go then to an introduction into immunology, because if we talk about infection biology, we need to talk how the body can fight against the microorganisms. Of course, it will be a very short section. If you want to learn more about immunology, you should choose immunology in the fourth semester or so. And we will talk about vaccines. And in the case of the vaccines, I will focus again on potential vaccines against coronavirus. And in this context, I will talk about how does a vaccine work, and how what are the challenges and directions in vaccine development against coronavirus. And that's kind of the same as we did for the drugs themselves, to have a critical appraisal of the current developments, and that you have an idea how a scientist looked at that, coming directly from the field of vaccine development. I did in my life vaccine development against viruses, against bacteria, and against cancer. So therefore, in this field, I have a real experience. The other fields, I'm not unexperienced, I would say, but in this field, that's my core experience. Okay, but now let's start with infection biology. That's what you should all know. And now let's have a look on the introduction, and let's see what it's all about. Now, what is the relevance of infection biology? So this is the situation in the States. We have at current 2004, it's not really changing. So at current it's more or less the same. So you see that the vast majority of people died due to cardiovascular diseases. There will be an increase now with of the infectious diseases due to corona, but it's if you compare it to the other diseases, it will still be marginal, even though in US they have still not stopped the disease going on. By the way, also in Germany, if you now go down with contact measures, then you could get back into the trouble. This is quickly made, but you see that the vast majority of people die due to cardiovascular diseases and cancer together. So this is more than one half. Yeah. And now, of course, if you come with chloroquine, for example, which has cardiovascular side effects, you increase that number here. So that's the problem. But of course, if you go to other countries, things are different. So if you go to Africa, so it's complete Africa, you see that 56% of the people still die due to infections. And you remember there, we have, for example, the Melinda and Bill Gates Foundation. This is the main foundation still supporting research against tuberculosis, malaria, and so on. So these are still the big killers here. So without this money, the research in this field would be very, very small. So the funding, unfortunately, doesn't really come from the rich states. It comes from private fundings into these I don't want to say neglected diseases, but diseases which are mainly affecting developing countries. So there is still a lot to be done to bring these numbers of these infections down. Yeah. And deaths due to infections down. So the relevance is high, although of course in industrialized countries it's lower. However, of course, with the current corona disease, every politician now is aware again of these infections, and I'm quite sure that the money which goes into this direction will increase. But just after having a disease which also affects the industrialized world, and not because of looking at this sad figure in Africa, where you have still in 2004, and that's of course not. I didn't update the figure here. It didn't change too much now for 2015, 2019. It's still the majority of people die there due to infections. Now, just to start with some definitions. There's just words you need to know. If people talk about infection biology and so on, you need to know what they talk about. We will have a homework also where you have a paper, and you will see that if you read papers, it's quite annoying if in every sentence you have to look up 5 words. So it's important that you have, let's say, some basic knowledge of definitions. What is a host? The host is the organism harboring the pathogen, and the pathogen is just the organism living in a host and causing a disease. If it doesn't cause a disease, it's not a pathogen. Yeah? An opportunistic pathogen is an organism which causes a disease in a certain host, usually an immunocompromised host. So there are many pathogens which, if you're healthy, don't do anything. So remember, coronavirus often does not provoke any diseases. So it's not an opportunistic pathogen because it's a virus. It will need to replicate and so on. But bacteria, you have often bacteria which usually you don't see any disease. And if you go, for example, you have then seen diseases in HIV patients which were immunocompromised, and then these pathogens developed a disease, or patients who had an organ transplantation and therefore the immune system needs to be suppressed, they then get disease. That's an example for opportunistic pathogens. Now, the pathogenicity is the ability of the pathogen to cause a damage in the host. And the virulence is just a quantitative measure of pathogenicity. The infection is the initial colonization of a host. So infection doesn't mean something evil. It means it's just you are infected with a organism, means that you're colonized by this organism. It could also be that if you're a girl, for example, and if you're newborn, you have an initial infection of your vaginal flora with lactobacilli, which is important for you in order to fight against, for example, fungal infections in the vagina, which is annoying. So therefore, infection is not necessarily something bad, although the term in a common way is usually used in a negative manner. And of course, an infection with a pathogenic organism, and that's usually what you're interested about if you talk about diseases, is something which is not good. Now we come to disease. Disease is, of course, the damage or injury of the host due to an infection, followed by replication of the pathogen, of course, or an intoxication with microbial products. So a disease means that an overt disease means that you realize that something is going on, that you develop symptoms. Now we go further, and this is a very important thing also now with corona, are these terms incubation period, latency period, contagious period, and subclinical infection. So the incubation period is simply the time between the infection and the first onset of symptoms. So here, this incubation period, for example, 10 days, and after 10 days, the disease starts. So if you are diseased, you have symptoms by definition. Now there is another term. This is the incubation period. We have the latency period. The latency period is the time between infection and infectiousness. The infectiousness is called the contagious period. And the contagious period don't necessarily needs to be the same as the disease period. And this is also most likely the case for corona. And this is, of course, something which makes us extremely unhappy, that you have people who did not yet develop symptoms, but they're infectious. And this is, then of course, it's very difficult then to, if you just tell as soon as you develop symptoms of a cold, like coughing and so on, stay at home, it might be too late. And therefore, in this case, for such a disease, you need this social distancing, you need this mask, and the RKI took way too long to recommend that to the people in Asia. People were way ahead with that because you protect others using these masks. So this is something you have to do there. And of course, this is if a pathogen has this behavior, this is very unpleasant for infection biologists and for the society, because this means that it's easily spread the disease. Okay, so that's about this. The incubation periods can differ tremendously from disease to disease. You see at the side here, you see leprosy. So here you usually have incubation periods of several years until there is an outbreak of the disease. Yeah, and usually, and this is of course then hard if you have these long incubation periods. It's hard then to realize what was the cause of infection. That was the same for AIDS, HIV infection at the beginning. Where does it come from? Because the distance between the infection event and the outbreak of the disease is so long. But there are also diseases where you have incubation times of hours. So for example, food poisoning with salmonella. So if you eat something which is contaminated with salmonella, usually you vomit and have diarrhea a couple of hours after this. And then you remember, oh, the sausage, I already thought during eating it that was kind of smelly, and then you know this was the cause of the infection. So you see many diarrhea stuff is here, but then you also see that you have other diseases where you have a longer incubation period. For corona, the incubation period is around about 7 days on average. So this is well in the range of other diseases. Okay, now the virulence. Remember. The pathogenicity is that a strain can cause harm, and the virulence is a quantitative measure of that. So you need to have ways to do this measurement, and one way to do that is an LD50, assessing the LD50. So what you do here is, in this case, LD50 is the lethal dose, where 50% of the animals die due to the pathogen. So highly virulent organism like Streptococcus pneumoniae. So if you infect with just around about 50 colonies or 50 living organisms, usually this will be IV, then you already kill the mouse. You can also use an EC50, ED50, and so on. So a dose where you have symptoms, but this is then the lethal dose. And if you have an organism like Salmonella enterocalcifera typhimurium, you see that the LD50 value is in the range of 10 to the 3, 10 to the 4. So it's less virulent. So with this LD50, you can assess the virulence. You have the same value for assessing the toxicity of substances like botulinus toxin. You know that this LD50 value is very low, and you can do that the same with pathogens. I have done such an assay only once. I had to do that once because we have created a new strain and we needed to know the LD50 value. For animals, this is of course horrible because they die due to an infectious disease. This is not something what makes the animals happy. Of course, due to animal protection laws, you cannot wait until the animal dies to it. You have to observe them very closely, and if you see that this animal will die within the next 2 hours. You have to kill it before it's dying due to the disease. Nevertheless, these are experiments which are, of course, extremely painful and for the animals. It's not necessarily pain, but of course they die due to infectious disease. They feel very sick, and this is so. Therefore, of course, you avoid these type of experiments each time when it's possible. Okay. Now let's come to virulence factors. Now we have talked about just basic features of definitions for infection biology, incubation time, and so on. And now we look at the organism, and now we look, but what actually does make a pathogen a pathogen? What is the difference between a pathogen and a normal bacterial strain? Not all bacteria provoke a disease, so what does do it? And maybe some of you, I think, with the corona crisis, the world has developed to infection biologists, but I still recommend this game. Yeah. Now it's kind of almost cynical if you play it, because now we have an infection which let's say the complete world is really affected, severely affected. But nevertheless, I consider this game as quite good, also for students in infection biology, because this really tells you what matters with respect to infection and virulence factors, and you can learn quite a bit out of this game. Okay, and I think one thing what everybody has learned out of this is that in a plague Inc, you remember you have your DNA points, and you can then maybe what we do is yes, I want to know it, so therefore we just do it. So I'm going back to invote. So no, that was too early. Just give me a second. This invote makes me so the problem with invote is that it kicks you out always, and the problem is that I need to use the Chrome browser because of the live stream, and the Chrome browser doesn't remember my so okay. So now what I'm asking you is, it's the same URL as before. So I want to know from you whether you have already played Plague Inc. or not. So who knows this game and who has already played it. And just, again, you can scan this code. I'm hiding my superbug, and just scan the code and tell me yes, no, whether you have played Plague Inc. or not. It's the same link as before. Okay, coming back to this slide. Now, I think all of you are aware that it's not enough to have just one difference. Yeah. So pathogen must have. Now you can't see anymore the. So the pathogen must have several different capabilities which are provided by different factors. And that we have the definition: a virulence factor is a molecule produced by the pathogen which contributes to its pathogenicity. So it's very simple. So that's where this comes from. Now I'm just looking whether there are already the first results here. So this should... no, I didn't start it maybe. Oops. I didn't start it. Wait a second. Again, a second. Yeah. Tuck. Generic question English. Oh, it started. So it should be here. Yeah. So the new address. And let's get it here. And let's go to the presentation mode. Okay, so 40% at present, 15 people have participated. 40% roundabout know this game, have played it. 60% don't know it. So it's just a recommendation. It's free. So it might be a wise idea just to play it one or two times that you have an idea how this virulence factor and so on, the principle looks like. It's a good way to get it. But now we have Corona, so you have unfortunately now the possibility to watch a disease appearing in real life, which is, I would prefer that we only have the possibility to use that game. Okay, just to see whether something has changed. No. Okay. Now let's go to the next slide. This is how it looks like, by the way, in this Plague Inc. And you see that you can, that's of course the German version, that you can then make symptoms and so on. And then you can your bug mutates, and then you can buy with these mutations new symptoms. Yeah. By the way, I played that on Saturday, and my wife told me, "Are you playing? You said that you prepare lectures." And I said, no, I'm preparing lectures. I need to do that because I need a screenshot in order to present that at the lecture. So I was in trouble with my wife due to this lecture, believe it or not. So this is because it looks like I don't work. That's of course not true. Okay, maybe just coming back to the chat, whether there are new questions. Oh, here are a couple of questions. Uh, as an American, you can call Trump a liar and idiot. It's okay. Yeah, I know. That's the usually I'm quite. I'm not an American. And can you talk so about other people? I was very, very, let's say, I had my opinion and never told my opinion. But in this crisis, this guy kills people. If you stand there and say, inject disinfectants and 20 people die to this is something which is completely unacceptable. So this is something I think which is absolutely it's absolutely incredible. For me, I was really shocked in this case. And this is then the time where you really say, this it doesn't work. Yeah, and this is the point. I don't blame, by the way. I think it's important for the Americans here. I don't blame the states. I know that I've been there. I have many friends there. That the people are really divided. That you have the supporters and non-supporters. It's really a sad story to see that. But at this position, if you see that a president of the United States tells people, and it was not a joke at this time. Use the scientists should try to use disinfectants to inject, and he should know as a president that people will do that and try that. This is something which is completely insane. That's really a point. So this is a point where you say. This is from my science. This comes from science, so I don't talk about other things where you can debate. This is science, and this is completely unacceptable. It's not that I'm so happy with other presidents either. So you could debate of politics over hours, but others. So Bolsonaro, for example, said, "Yeah, it's no problem." Also in Germany, we had at the beginning, people said, "Oh, everything will be fine," where the hints were different. In China, they hide everything. So I don't want to say that Trump is the only evil person in the world. Please get me right. There are many things which make me unhappy in this context, but he was the one who said use disinfectants, and that's not a good idea. So infection is a single moment in time rather than a period of time. Yes. Yeah, it's not necessarily that it's just a single moment. Usually you are exposed to the pathogen, but this exposure period could also be longer, but it depends on the infection. Most offended patients on antiviral get diarrhea. Is it because the system is immunocompromised? That's a good question. Honestly, I don't know. So this could be that this is just a side effect of this antiretroviral drugs. I honestly I don't know. This could be a side effect. I'm not sure. Yeah. I don't think that it's due to the immune compromised system, because diarrhea is for drugs who are, let's say, more have more toxic side effects, and retroviral antiretroviral might have a side effect. It's a quite common side effect. So I think it's a side effect of the drugs and not something with immunocompromise, but I'm not sure. Greenland for the win. Okay. Plugging. Is LD50 aware of the numbers of injected pathogenic cells? Yeah, so the LD50, at the end, the LD50 is the dose. So the LD50 is the dose, for example, 500 bacteria you have injected, what you need to kill 50% of the animals. 50% means, of course, it's not just one animal in the experiment. For LD50 experiments, you have different doses, and for each dose, you have to have at least 5 to 10 mice. Yeah? So you have, in such an experiment, if you already know what doses you need to apply, you have 50 mice in the experiment. So these are not so small experiments, and really not nice experiments. Can he be prosecuted based on the claim he did? I hope. There's a possibility, but mostly because he's restricted medical relief to certain states. He didn't like the mayors. Yeah. Yeah, for me, honestly, if someone tells you inject disinfectants and kills people, well, if I would be the relative, I would sue him, of course. But, well, okay. Okay, so but now I want to stop this this discussion. We need to go back to microbiology. I'm quite sure that the people, that I'm very happy that the people studying at the Rhine-Waal University come from all countries, and it works. And that's also because usually one should be quite moderate with respect to political discussion. So I'm sorry now that we went a bit over the top. It's just because at the present I'm just shaking my head, and I had headaches because I needed to shake my heads in this case. But one should be tolerant, and there is a difference between the president and the population. Being in the States, and therefore I have to make an advertisement, so Running All Day will confirm that. The people are so nice in the States. Everyone, you go there, and it's enjoyable if you talk to the people. And Ralph, who is in the background, will confirm that. It's completely different to Germany. In Germany, people are always negative. If you talk to people, it's always like, oh. And this is completely different in the States. So I like this country very much. That's just to make that clear. Yeah. Okay, now we go back to our presentation. Again, I said as we had to start 50 minutes later, we will go until 10 o'clock, but then we will stop. It's not that we will go until 10:30 like last time. Now, what are the basic steps in the virulence? And so this is how an infection occurs and how the disease at the end breaks out. So first of all, you need an exposure to the pathogen, and then you have the adherence to the skin or mucosa. So exposure, that's important. Exposure, it's not enough. The pathogen must somehow stick to the site of infection. If it's just there, and if you make like this and it's gone, there won't be an infection. So the pathogen must have the opportunity to stay there. So there is the adherence, and you will see later on that for each of these steps you need then virulence factors in order to achieve that. Then you have the invasion step, and then you have the colonization and growth, and then at this step, you might have a further exposition at the local site, which re-exposes you again, or you could have an invasiveness, and you have other sites get exposed. So a local site means that this is a typical of the smear infections. If you have a streptococcal infections, for example, in the face, kids have that very often, and you scratch, then you have exposure at other sites, and then that turns bigger. Now, if you get into the systemic side, then you might have have an infection of the liver also at distant site. That's further exposition at distant site, and this only happens after the organism has invaded and there is a growth at original and distant site. And you can of course also have the toxicity, so toxin effects, which are local or systemic. Toxins, we will talk about that of course in more detail, are substances which are produced by the bacteria and by themselves cause effects which are not due to the growth of the bacteria. And at the end, you will have a tissue damage or overt disease. So that's what you realize. If you just have some bacteria growing in your skin and there is no immune system, no toxin, nothing, then you won't realize it. So this effects here then make your tissue damage and overt disease. So that's of course a very basic but a very important slide which summarizes what all must happen in the case of bacteria. Now, in other words, that's just the same, just in another presentation, and you see that you have these different portals of entry, and then of course it's a function of the number of invading microbes. It makes a difference whether you're exposed to just one organism or to 1,000. So that means that wearing that mask is smart, because the exposure level of another person will be reduced. But depending on the disease, if you have highly infective diseases like measles, mask or something like this will not help anything, because it's enough if you already see one bug you were infected. Yeah. Then you have penetration, damage to host cells, and portals of exit. This is something you can read also at home. So these are the different things which confer to that, and we will talk then later in more detail about these substances. Okay, so I would say the chapter adherence capsule CD4 we do today, and next time then we will start with toxins. Why we can do that? Because it's a very short chapter and there is not so much to be said. Toxins is certainly a larger chapter, and we take that then for next week. Now, adherence, if we look at that, we have bacteria like pathogenic bacteria, of course, like Vibrio cholera, of enteropathogenic Escherichia coli strains, and you see in this case it's the microvilli layer in the intestine. You see that they're closely adhered to that. So you see this is an electron microscopy, and you see that they come very, very close. And this means it's not just that they approach it. If you're so close, if you see this distance in an electron microscope, it means that there are factors which attach you to the surface. This is adherence. So it's not just that you somehow because you have a certain mass interacted with it, you really need to attach to the surface in order to stick to it like this. And of course, if you want to make an infection via the colon, so an oral propagation of infection like cholera or so, if you cannot stick to the mucosa in the colon, you're just washed out. So with a normal stool, you're gone. So you need to have the opportunity to stick there, otherwise you're eliminated. And an example here is just again this enteropathogenic E. coli strain. So here this strain has two types how it attaches. At the first step, it's a loose attachment via a pilus to the host protein BFPA. And then this E. coli strain establishes a very strong adherence. And this is done that this bacterial strain produces a protein called TER. And this TER is type 3 secretion system translocated intimine receptor. So it brings a receptor into the cell, and this then can interact with the intimine on the surface. So it injects a protein into the host cell, which is then exposed on this microvilli. And then you establish a very, very, very strong attachment, and you have no chance at all via physical means to get rid now of this bacteria anymore. And you have three things which are involved. This is the intimin, which is in the surface of the E. coli. You have this TIR, this type 3 secretion system translocated intimin receptor, which is exposed on the surfaces. And you have a type 3 secretion system, which actually injects that into the cell. So you have three factors which are important to establish that attachment already. And now, if this is attached, you have remodeling of the actin skeleton, and you have so-called pedestals. And you see that you have a disruption of the structure of the colon. Of course, you don't have just one bacteria, then you have several ones. And of course, there you distract the microvilli, which leads to an inflammation and death of the epithelial cells. And at the end, you lose electrolytes and have diarrhea. So you end up with diarrhea in this case. Now, this is something I want to skip because we are already a bit far away. Of course, just adhesion is certainly not enough. And now I just have a list of virulence factors examples with relevance for initial adhesion. This is also something I will not ask you. It's kind of give an example for a pathogenicity factor for adhesion, and we have already talked about that. So if you give this example, everything is fine, but I just want to tell you it's not just that. You have different pathogenicity factors, different categories here: motility, adhesion, and you see that they are present in different strains. Interestingly, you find the E. coli strain UPAC. This is the uropathogenic Escherichia coli. ETEC is the enterotoxigenic Escherichia coli, and EHEC is the enterohemorrhagic Escherichia coli. So that's all E. coli strains with different virulence factor leading to a different strain. Now, another virulence factor is the capsule, and this is the kind of the classical example is Streptococcus pneumoniae. So you have two forms of this strain, which was already known long ago. A strain which forms rough colonies, and a form which forms smooth colonies. And the cause is that these smooth colonies, these strains, have secrete polysaccharides. Therefore, they form a polysaccharide capsule. And of course, such a polysaccharide capsule around the strain leads to the embedding of water, and therefore you have this smooth drop-like colony form, contrast to this rough form. Now, the difference is that the immune system can easily recognize this R strain because the surface, the cell wall of this bacterium is easily accessible to components of the immune system. In contrast, for the R strain, it's not accessible. So for the R strain, the problem is that due to this capsule, the immune system cannot attack this strain, and therefore it prevents the detection of host immune system. And that's a very important thing because that makes changes between life and death. And this is then this Griffith's experiment, this very, very famous Griffith's experiment, where he demonstrated DNA transfer in this experiment, bacterial transformation, and which was a milestone experiment here. And he used animals to show that, but it was important for the people to believe that. I would say someone of you can explain that to me next time when we talk about that. And finally, siderophores, that's the last slide, or the last topic here. Siderophores are small molecular substances which can bind iron and take up iron, because in many hosts iron is limiting, and therefore you need to have access to iron. And these siderophores can complex iron with a very high affinity, so you can take away the iron from the host, and you have an active uptake mechanism. So in order to be able to grow in a host with these low iron concentrations, it's important for the bacteria to have this siderophores. This siderophore, by the way, which is depicted here, is not from a pathogen. Also in soil, you have low iron concentration, so many soil organisms also express siderophores, including strains like Streptococcus myces. Okay, this is something I can skip. And then we are at the next chapter, and I promised that this is something we will do next time. So I will look again if there are more questions have came up. Okay, so I think no more questions. So sorry again for the beginning. So next time, if I have this, I hope that I don't have this problem again, because I will not click and change the stream rate at the same time when I start the stream, because I think that was the problem. But what I will do if the stream doesn't work, and it doesn't work, usually I start the stream really at 8 plus minus 1 or 2 minutes. If you see that the stream doesn't work, write it in the please write it a short message in the chat, because the chat, as you have seen, worked already. And I will set up a new, if I have to set up a new stream, I will put the address on Moodle, because the problem is, as soon as I stop the stream again, the chat is also gone. This is my problem, and therefore having two streams ongoing is quite complicated for changing the streams and so on. It doesn't work really good. So then I can stop the stream. I will then write in the chat, I will stop the stream, and then you know that the new URL will be on Moodle, and this is a faster way than, and I don't need 10 minutes, and maybe it takes just 5 minutes for the new stream to go. So thank you very much for your attention. This time we were overall in time, and I hope it was not as dense as last time. Looking forward to seeing all of you next week. And well, next week I don't know how far we go, but maybe next week we can already talk about vaccines, and we can talk about vaccines against coronavirus, and what are my predictions in this case with respect when we will see a vaccine. So thank you again for your attention, and have a nice day. Goodbye.